And Dr. Dhanker served as a research associate prior to coming to GD Goenka University at MIT University, research assistant in Central University of South Bihar, and Acharya Narendra Dev College, Delhi University. She has qualified national level life science examination, so-called JRF and GATE, and published many articles in SCI referred journals. Besides, she has written few book chapters that was published in Elsewhere and Springer Publication House. Her current research interests are in the area of environmental biotechnology, green nanotechnology, and aquacultures. She served as a reviewer in a journal of plankton research and PLOS 1. She is a frequently presenter in the national and international conference. She has delivered several invited talks in various national and international conferences, presented a paper, and received several times Best Presentation Award, Poster Award in national and international conferences. Under her, she is also supervising a Ph.D. students, and students have also done quite well. So she has a total more than say 7 years of teaching and research experience. Dear colleague, today's session she will focus on bioprospecting. The other name is biodiversity prospecting. Is the invention of biological material for commercial value genetics and biochemical properties. The purpose of microbial bioprospecting is to discover new resources and products from living beings, from commercial benefits of human, as well as to enhance research possibilities, economic development in a sustainable manner. The main focus of microbial bioprospecting is to generate clean energy, improve human health by medicine, as well as to get a sufficient amount of nutrition from microorganisms. India, one of the largest biodiversity-rich country, can get benefit from bioprospecting by promoting education and research on their own biodiversity. Dear colleagues, now I would like to invite Dr. Dhanker to deliver this talk, and prior to that I may kindly request you to mute your mic and camera, and also you can place your question on a chat box. Our dedicated faculty members led by Dr. Renu Chaudhary and team, they will ask question once the resource person will complete the talk. Thank you so much indeed. And Dr. Dhanker, I would like to invite you to start your talk.
Thank you, sir. Very good afternoon to all. Actually, I'm facing problem in presenting my PPT. So, because my laptop is not properly working actually. So, it will take few seconds. So, I will start sharing my PPT now.
Good. Just go on, please. I have some obligations, so I will join you later on. Thank you.
Okay, sir. Okay, sir.
Yeah.
Fine.
Thank you. Please go on.
Renu ma'am, is it visible? My PPT is visible?
Yes, ma'am, but you have to go to PPT. Your screen is visible, your Google Meet.
No, ma'am, your PPT is not visible.
Yeah.
We can see presentation, but your PPT is not visible. Now I think you're presenting, no?
Yeah, I'm trying actually my laptop.
Or else you can mail someone. Anyone else can present on behalf of you.
Yeah, it's opening now. I think.
Yes.
Yeah. Yes. I hope it is visible now.
Yes, ma'am.
I'm audible?
Yes, ma'am.
Yes, ma'am.
So today here in the session of STB series, in the 7th session of STB series, I'm going to discuss about bioprospecting of aquatic microbial community. So life on Earth was started before 4.5 billion years ago, and from during that time till now, the natural selections has shaped the organism community and their chemical constitution in a very complex way. So organism can release huge amount of biological active natural products which are really impossible to be synthesized chemically. So what is bioprospecting? So bioprospecting is also known as biodiversity prospecting, and biodiversity is the exploration of natural sources for small molecules, macromolecules, for biochemical and genetic information. Due to that, the many value-added products can be formed for the benefit of human beings. So more than 90% of drugs, at the first place, I want to clear that bioprospecting is not directly related to pharmaceutical industries. Bioprospecting is related to every kind of wealth which we can generate from the microbes. But in the past, the old definition, it was focused on this term is related to drugs. So in past, more than even today, also more than 90% of drugs was directly synthesized by plants. But later, due to the technique of recombinant DNA techniques, many wild genes were explored and they were cloned in the respective organisms for getting more beneficial products. So bioprospecting is related to their utilization in agriculture, aquaculture, bioremediation, nanotechnology, pharmaceuticals, and many more industries. So these are some major areas of bioprospecting, which include like chemical prospecting. Under chemical prospecting, we study about drug and pharmaceuticals, pesticides, cosmetics, food additives, gene perspectives, in which basically we study how we can manipulate the genetic information of any organism so that we can get desirable product, we can improve crop. So this application will include in agriculture. Then bionic perspective, in which we design many sensors, and bioengineering and biomodeling techniques come under this area. So there is a difference between exploitation and exploration. When we say that we need to explore many natural products so that we can get many value-added products for the benefit of living beings, then the generation of revenue come into the picture. And when the generation of revenue come into the picture, definitely we need some legal aspects so that we can control the utilization of these natural resources. Otherwise, the exploration of natural resources will convert into exploitation. So that the native country can get the benefit of these natural resources, some conventions was formulated. So in this regard, first convention was formed in 1992 during the Earth Summit in Rio de Janeiro, which was till today, which was come under the category of largest convention, because more than 193 countries was participated in this convention. And this convention was focused on three goals. First was conservation of biological diversity, and second was that how the utilization of this biodiversity can be done in a sustainable manner. And last was, but this was the most important legal aspect of this convention, that the fair and equitable sharing of the benefits from the use of all these resources. So later in 2010, another treaty was come into the picture, and the name of that treaty was Nagoya Protocol. And this treaty was also an international treaty And in this treaty, approx 96 countries were participated, and the main purpose of this treaty also was to sustainably utilize the biodiversity, and legally the benefit of these natural products can be taken by the native countries. So when we discuss bioprospecting, two kinds of habitats come in our minds in which microorganisms are found. First is terrestrial ecosystem and another one is aquatic ecosystems. So terrestrial ecosystem, like we include deserts, like plants, forest, and herbs and shrubs. So in these categories, plants basically utilized as a bioprospecting products mostly till now, but comparison to terrestrial bioprospecting, aquatic bioprospecting is done in a very less way, or it's still unexplored. So when we say aquatic biodiversity, it means the living beings who lives in different aquatic ecosystems, like the microorganisms which live in estuaries, which live in fresh waters, which live in marine ecosystems. So marine basically terms related to salinity, salt concentration in the water. Generally, the salt concentration on marine water is more than 35 ppm. And basically fresh water has less than 5 ppm. And estuary is a habitat which is the transaction zone of freshwater and marine environment. So aquatic biodiversity, we need to select marine as well as freshwater microorganisms. In these categories, we can include fresh fungus, bacteria, the microorganism which lives under extreme habitats such as archaeobacteria and microalgae. So basically, the technical details of bioprospecting is like, first we need to identify which microorganism can be more beneficial for getting natural products. Second, after identification, we need to culture those species in laboratory. After culturing, we need to do screening. Screening means which species can be more useful and which can be less useful. Then, is there any ethical issues or safety issues related to that species? The main purpose of these natural products must be that how these natural products can be utilized further for large scale commercial production, so that the country can get benefit from these natural products and the economy of any country can be raised. So during my talk, basically I will focus on algae. And why algae? Because algae constitute more than 80% of aquatic habitats. They are primary producers, means they can produce their own foods. They are the backbone of aquatic food chain. They are eukaryotic. Most of the algae are unicellular organisms, but some algae can be multicellular also. They come under the category of seaweeds. So algae further can be classified in you can see in your around green algae. They come under chlorophyta. Another category of algae is brown algae. Then there is another different category of algae is red algae. They come under the category of phaeophyta. So algae basically are found in every kind of habitats. They found in both lantic and lotic habitats. Lantic means that the water which is stagnant, and lotic means running water. So algae is found every kind of habitats. Even their presence can be identified in desert also. So after collecting algae in the laboratory, or we can culture algae in like outdoor pool also. In scientific language, or those pool we say mesocosms. So we can design mesocosms and we can culture microalgae out of laboratory also. In laboratory also we can culture them in flask and beaker also. So after culturing and screening and after checking their efficiency, the algae can be utilized in different applications. They can utilize for industrial applications, such as many nutritional products can be extracted from the microalgae. They can utilize in nanotechnology, because some species of microalgae which come under the group of diatoms, their cell wall is form of silica. So they are a very good vehicle for the drug delivery. And after the death of diatoms, they release silica in their environment, which is used as like in other words I can say, which is a beneficial death, because that powder form of silica can be utilized in agriculture purposes as an insecticide, as a herbicides also. In pharmaceuticals, the algae has many biomedical applications. They act as antioxidants. They are prominent source of bioenergy. So they can be utilized for the production of biofuels and biodiesels. And their residual part after the extraction of biofuels can further utilize as biofertilizers. So algae can utilize wastewater, and because they synthesize their food with the help of photosynthesis, so they consume excess amount of carbon dioxide from the environment. So like this, they also help in bioremediation. They also act as bioindicators, means they can tell the condition of any aquatic habitats, is it eutrophic or oligotrophic, because algae can access extra amount of nutrients also. They have their role in forensic science also. So algae is basically, besides producing many valuable products, algae also synthesize many secondary metabolites. Secondary metabolites means the products which cannot be directly utilized by the algae for their growth and reproduction. So due to these secondary metabolites, they shape the population community in aquatic habitats. So like this, they open the possibility of discovery of new drugs for pharmaceutical industries. So how algae can be cultured? So this image is showing different techniques which can be utilized for culture and harvesting of the algae, so that their biomass can be utilized for the extraction of value-added products. So first we need to collect algae. After that, if we want to culture them in fresh water, that is also fine. But if we want to culture them wastewater, that is also an extra advantage. This is known as integrated approach. So integrated approach means at the one side, we are growing algae for the utilization of human benefits, so that we can get many products from that. And on the other hand, we are purifying that wastewater. So basically, if we want to culture algae outside of laboratory for their commercial utilization, we can culture them in open ponds, in closed ponds. We can also culture them in photobioreactors. Or after that, we can utilize algae for the treatment of wastewater. So harvesting, basically, this utilize filtration, centrifugation, sedimentation process. So after this process, the algae, these are the pigments or substances which we can get from algae. So macromolecules, lipids, carbohydrates, and after extracting lipids and carbohydrates, their residual biomass can be further utilized for the extraction of different pigments, such as omega-3 fatty acids, exopolysaccharides, pigments, sterols. And omega-3 fatty acids, which is the most important form of lipids, which is utilized as a beneficial food for the secondary trophic labels, which include DHA and EPA. And other pigments are fucoxanthins, which have their application in antioxidant activities, beta carotene, astaxanthins, chlorophylls. So if we say algae as a biorefinery, means the microorganism which can be utilized for the more than one purposes. So algae, as I told in my previous slides, algae can utilize excess amount of nutrients, carbon dioxide, and in the presence of light and water, the higher biomass of algae can be generated, which can be further utilized for the generation of biofuels, bioproducts, and other bioenergy products. So from onward, I'm going to focus only on biofuels and why we need to use alternative source of energy. Because as we know that there is an insecurity between there is an insecurity with the existing fossil fuels, conventional fossil fuels, because there is a one reason is there is a geopolitical instability. Second is for fossil fuels generation such as coals, there need like many years to generate that. So the reservoir is being getting reduced day by day. And another aspect is the agriculture land is lands are shrinking day by day. So instead of algae, plants can also be utilized for the generation of bioenergy, especially biofuels. But plants need agriculture land for cultivation. So that's why in comparison to plants, algae is better solution to get bioenergy comparison to plants. Another is from algae we cannot get only biofuels. We can get many other value-added products also. So that's why algae is better alternative energy source comparison to plants and conventional energy sources. So these are few parameters why algae is important comparison to another sources. All these sources I already have explained in my previous slides. So I can escape this slide. So how the history of algae as a biofuel was started. So there was a project which was carried out by US Department of Energy during 1980s and 1986. And actually this was an umbrella scheme. And this department launched the Aquatic Species Program. And during this program, more than 3,000 strains of different microalgae were tested for biofuel efficiency of microalgae. So out of these all strains, 50 strains were has shown outstanding performance for lipid productivity. And surprisingly, out of these algae, 60% was diatom. And diatom is a brown, golden brown color species which has unique attribute comparison to other microalgae, because diatom has silicated cell wall. So that's why they are unique. So there are some other attributes also of diatoms which are unique comparison to other microalgae, such that they can utilize excess amount of nutrients more efficiently comparison to other microalgae. So there are some factors which can influence the bioprospecting of algae. So when we discuss about factors, they come under two categories: biotic factors and abiotic factors. So abiotic factors are light, and when we talk about light, two factors come in the picture: quality of the light and quantity of light. Another is pH, nutrients. Nutrients basically nitrogen, phosphate, and potassium. Then ultraviolet rays. That under ultraviolet rays also we study different types of ultraviolet rays: temperature, fresh culture or old culture, aeration, turbulence, then salinity. And the biotic factor is predators, which graze on microalgae. Then another factor is mechanical bonding, which release chemical compounds in their environment. And last but not least is different types of the biofuel producing capacity of microalgae is also depend on different types of species and their strains. So what are the challenges? If we want to use microalgae as biofuels, there are some challenges. Such as first, as far as I also have studies many literatures, most of the study has been done in laboratory only. So there are very few scientific evidence which really show that algae can be utilized at commercial scale. Another is very limited information is available on strain specific productivity of algae. And some algal species such as diatom, they require additional media. So in their media, they want silica for their growth because they have silicated cell wall. So basically when we want to culture diatom, we use silica F2 media. But if we want to use media for other microalgae such as green algae, we can use multiple media such as BZ11 and Cullen-Lorenz media and simple F2 media. So if we want to culture diatom, so additional cost is required. And another important factor is because when we culture microalgae in an open area, because environmental factors, biotic and abiotic, vary there. That's why it's very difficult to get an appropriate amount of biomass. Because when we culture them in open area, in the starting that will be contamination free. But slowly what happened? There will be an emergence of predators also. So that's why that factor will also reduce their biomass. And another, so how these kinds of challenges can be mitigated. So one of the example of that is we can use an integrated approach. So in integrated approach, what we do, like as I discussed earlier, algae utilize carbon dioxides and excess amount of nutrients. So they can be grown in wastewater. So after that, these biomass of algae can be extracted for the production of many forms of lipids, which can be further transfer into biodiesel and other forms of fuels. And the residual form of this algal biomass can be utilized for the production of different value-added products. So like this, we can treat wastewater also, which can be further utilized for domestic purpose and agriculture purpose. We can reduce carbon dioxides from the environment. We can reduce excess amount of nutrients also from the environment, and we can get many different types of value added products also, and bioenergy also. So I thanks to ZD Goenka University for providing me opportunity to deliver this talk. So this is some awards and ranking which we provide to our students in our university. So student is also exchanged for research and semester study in abroad. So these are different schools in GD Goenka University, and we are the part of School of Basic and Applied Sciences. Professor Dean Ramkrishnan Thakur, he is receiving award during the MCMCCI Green Tech Asia Award. So these are the 6 faculties of Department of Physics which have different research interest: ultra cold molecules, then spectroscopy of rare earth molecules, supercapacitors, then Department of Chemistry, which include 8 faculties, and faculties has very good research experience or teaching experience from good reputed Indian universities and international universities. Mathematics departments, total 5 faculties. Biological sciences, 3 faculties, which are doing their research on drug delivery, green nanotechnology, and cancer therapy, and other area also. Department of Forensic Science, total 5 faculties, which are working on forensic medicines and signature identification, forensic toxicology, and other research areas. So right now, 3 research projects is going on in SoBAS, which is funded by different funding agencies, including DST, Government of India. So the picture is showing the different facilities of laboratory which we provide to students in our school. So these are programs, undergraduate programs, which we offer in SoBAS: chemistry, physics, mathematics, forensic science, microbiology, biochemistry, biotechnology. We also offer different types of BSc Pass courses. After doing undergraduate, student can go for master, or they can work in different companies. So during postgraduate, we offer these different programs: MSc, chemistry, physics, math, forensic sciences, microbiology, biochemistry, biotechnology, data science and scientific computing, geology, wildlife, forestry, biophysics, environmental science and management. Student is also doing Ph.D. under the guidance of SoBAS faculties in different areas. So these are diploma courses which we offer in our school in criminology, cosmetics, data science, environment, health and safety. So for enhancing the learning and teaching process, so we have, like, student first can interact to mentor. If the problem is not solved, they can move to SOD, and if still there is some doubt, student can reach to dean. So for enhancing learning experience, learning process, time to time school conduct educational trips to industries and laboratories, conferences, faculty development programs, student development programs, workshops, symposiums, and many expert talks also. So here we can see in the pictures Professor P.B. Sharma, who is the founder of many Indian universities in India, and right now he is working as Vice Chancellor in MIT University, Manesar. And Dr. Kumar Krishnan, who is NASA scientist, who is also recently delivered his talk during the Eminent Scientist webinar series. So these are few glimpses of experiential learning in which we invited Dr. D.K. Aswal, Director of NPL New Delhi, and Professor Ramkumar, who is Dean in Central University of South Bihar. So another is Dr. N.B. Burdan, Director of CBI, CFCL Delhi. So these pictures is showing some glimpses of student development programs. And this image is showing that student is visited to industrial trips in Delhi University, NPL, IIT. And students also visited to AIMS and participated in Anveshana program. So in 2020, online conference is also conducted, and GD Goenka SoBAS is also organized international conference before lockdown. So outreach programs. So for outreach programs, time to time school conduct many school conduct program. We also invite school students in our school for their interaction with the faculties, or they can get experience in our school. So these are some training and placement companies where students are placed. And during the lockdown also, SoBAS is successfully conducted career counseling webinar series and eminent scientist webinar series, which is still going on, and student development program series is also going on. So our next event is Global Water Congress that will be conducted is in like on the occasion of Mahatma Gandhi's birthday. So I will request to participate in this conference, so they can get benefit, they can get more benefit how the environment is working and how they can connect to the social activities through this conference. Thank you.
Well, thank you, Dr. Dhankar, for enlightening talk. It is highly informative for general point of view, and especially for the students of biological sciences, biotechnology. I'm sure all your talk must have somehow pushed our attendees to think at a deeper level and spark their creative idea. That's how we build our ideas, by developing our understanding through the talk, discussion, lectures, and so many other experiential learning activities and classroom teaching, laboratory supervision, and so on. All our aim is to work together to make each and every day a meaningful, productive, and happy day. So thank you once again. And now we open the house for the question. So Dr. Renus Chaudhary.
Yes, sir.
So could you please read the questions of the audience?
Yes, sir. There are a few questions.
One after another.
Yes, sir. So first question is from Marvin Matthew. He has written, ma'am, why are algae better sources of biofuels in comparison to terrestrial plants?
As I already discussed during my presentation also, because if we want to utilize terrestrial plants for biofuel production, we need agricultural land. And if we will utilize agricultural land for the biofuel production, then many value-added products which we can utilize from the plants, we cannot get that. So this is the reason.
Okay, I think Marvin Matthew is satisfied with the answer. Marvin, are you there?
The student is not here.
Okay.
I think maybe
We'll go to the next question.
mic is muted, so he, yeah. He wrote yes, the answer is fine. Good. Thank you. And next question.
Next question is from Vinita Makkar. She has written very nice session, Professor. How we use finite element method in case of biofluid?
Actually, they are different method, and finite element method is one of them. So, but the finite element method is also have some their own limitations. In this finite element methods, we can culture microalgae in different types of culture systems. So after that, we can harvest them, and we can use different kind of solvents for the extraction of biofuels. And after that, their residual parts can be utilized for the extraction of different kinds of pigments.
Okay. Good. So next.
The next question is from Gunja Kumari. She has written, algae which is present at deep ocean, how they survive without sunlight?
Actually, there are very less, although negligible, number of species which we found in the bottom of aquatic habitats. Basically, there we found extreme microorganisms. So microalgae, most of the species we found only in the open surface area. But some microalgae, which are like heterotrophic, they can utilize the debris of the debris of that microalgae, which sink in the bottom, and from that they can get organic carbon source, and like this the algal species can survive. But I'm clarifying again, there are very few, very few, rarely you will get algal species in the bottom of aquatic habitat. And in the bottom we can get only like Professor Hans discussed during his talk. Yeah, in the bottom we can get only some extreme microorganism. Extreme means we can the microorganism which can survive in high salt concentration, high pH, and very high temperature. The one of the example of that is archaeobacteria. So in the bottom we will get those kind of microorganisms.
Good. So further question, Dr. Renu Singh. And by the way, Dr. Kamble and Dr. Sethi, could you please also keep eye on the question? We should not miss any question from the attendant,
Yes,
attendees.
Sure, sir. Sure, sir. Sure.
The next question is from... Yes, sir.
Dr. Chaudhary, could you please read another next question?
Yes, sir. Next question is from Deepa. She has written, how efficient is algae biofuel?
Yeah, if I say at a small scale, it is definitely efficient, because it has been seen during the many study that many algal species has 40 to 60% lipids, which can be utilized for the production of biofuels. But as I discussed during the challenges point, we are really very far from the commercial utilization of microalgae as biofuels. But at laboratory scale, it is possible.
Okay, good. Further, next question, please.
So next question is from Anjali Singh. She has written, what are the challenges with algae used as biofuel?
Can you repeat?
Yes, ma'am. What are the challenges with algae used as biofuel?
Okay, if we want to utilize algae as biofuels, first we need to grow algae so that we can, in actual sense, we can meet the need of the local peoples. So for that, we will have to culture microalgae at a very mass scale. And that is not possible because many agencies has tried to culture algae at mass scales, but they got contamination of different kind of microorganisms very frequently. And another factor is environmental factor because they are also vary in the nature. So scientists has developed some another techniques like photobioreactors. So photobioreactors are like some like a transparent pipe, very big size, transparent size. But so from that algae can be harvested because that is a closed environment. So in closed environment we can culture algae, and we can stamp on the yes, this extract is completely found from the microalgae only, but at open place it is not possible. First challenge. Dusra, because when we will culture algae at mass scale, there will be the presence of predators also. Second challenge. Third one is abiotic factors because they will vary. So this is the another challenge. And last one is because if we talk in true sense, the fossil fuels, it's true that they are takes long time for their formation, but still they can meet the demand of the local people. Algae we cannot culture at that large scale, but lots of efforts is going on all over, all around the world. I hope that some more techniques will come out. The another challenge is cost. If you want to culture algae at large scale, even in photobioreactors also, the cost is very high. And especially if I discuss about diatom, which is the most important category for extracting biofuels in microalgae. So because they need silica for their growth. So an additional cost is also come during this, during the culture of this species. So there are lots of challenges.
Okay, great. And Dr. Chaudhary, next question, please.
Next question is from Gunja Kumari. She is asking, what are the challenges related to provide a proper environment to grow our algae in lab?
Actually, very less challenges if you want to culture algae in a closed environment, which is Gunja is saying lab. So in lab we have closed environment, like we can use BOD, and if you want to purchase BOD of good quality, like BOD has 3 to 4 chambers, we can set different temperature in the BOD. So like this we can set the temperature, because laboratory is a closed environment, so we can set the pH according to our requirement, according to our needs, because we in laboratory we know that which pH and which temperature and which other factor is more appropriate for the higher growth of biofuels. So we can set according to our convenience.
Okay, ma'am. I think Gunja is satisfied with the answer. Next question is from Sania Yadgir. Yes, Gunja?
What about the sunlight? We also need a sunlight for growing our algae.
One of the factor, Gunja, which I discussed light, and in light also I discussed qualitative and quantitative parameters of light. So in laboratory we have light intensity chambers, we have culture rooms. So we have culture rooms for bacteria, we also have culture rooms for the growth of algae. So we set artificial light accordingly. So next time, maybe next trip, I will show in laboratory in Delhi University, there some professor is culturing microalgae there. So we will plan accordingly.
Okay, ma'am. Thank you.
Okay.
Yeah, any more question?
Yes, sir.
please.
Next question is from Sania Yadgir. She has written, what are most the useful applications of algae so far as compared to other biofuels, and also the known conventional energy sources?
Actually from biofuel, the most important application of microalgae, their role in aquaculture. They are primary producers. They can synthesize their own foods. Whole food chain depends on them. If there is no microalgae, there will be no food chain in aquatic habitats because grazers feed on them. Like on algae, zooplanktons can feed. Zooplankton is another group which is also some species of zooplankton can be seen by naked eyes, but most of zooplanktons can be seen by under the microscope. And basically, for that we use stereo zoom microscope. So, because algae is primary producer, zooplanktons will feed on them, and zooplankton is the food of fish. So fish will feed on zooplanktons. Some fish also can feed on microalgae because they don't they are not carnivorous, and some fishes are omnivorous. So omnivorous means those fish or microorganisms which can feed on both plants as well as on microorganisms such as zooplanktons. So the main important role of microalgae in actual sense come in aquaculture. Aquaculture means how we can grow fisheries department, how we can grow the number of fish. And we know that the many states in India and many countries, because the 70% of earth is covered by water. So there are many coastal countries and coastal state in India. So the livelihood of big size of population depends on fisheries. So main role of algae come in aquaculture.
Oh, thank you, Dr. Dhankar. Dr. Singh, Dr. Chaudhary, is there any more question?
Yes, sir. Two more questions are there. Sania has also asked, is algae more efficient than other sources such as wind energy or tidal energy?
Actually, this is a debatable issue. So right now, I cannot reach on the appropriate conclusion on this, because a big group of researchers, they said that microalgae is more efficient, but another group say that wind and solar and other source of energy is more efficient. So sorry for this.
Okay, ma'am. Next question is from Ankit. He has written, can algae survive only with moisture in vacuum?
Yes, they can. Because few species, they are highly adaptive, few species of microalgae, they are highly adaptive to harsh environmental conditions. So they can. Even they can survive in desert also.
Okay, ma'am. And the last question is from Gitanshi Singla. She has written, name the best technique for extracting lipids for biofuels production.
Actually, there are lots of methods, but out of these, the sonicator method is the best, in which we use ultraviolet rays for breaking the cells of breaking the algal cells. So when we will break the algal cell more efficiently, we will get the extraction more extraction of more molecules. So best techniques is sonicator.
Okay, ma'am. Thank you.
So that's all?
Yes, sir.
So Dr. Kamble and Dr. Sethi, is there any question we have left?
No, sir. We are not left with any question. Dr. Chaudhary has asked all the questions.
Okay, great. So you kept eye on that. Great. So yes. Here I have a bit, concern about one question. Recently, Dr. Chaudhary asked, it is, I think, Ankit, ma'am, can algae survive only with the moisture in vacuum? And Dr. Dhankar said yes. So my concern is, if the vacuum is very high, I think there will not be any moisture. So in my opinion, algae can't survive. It could survive in a low vacuum. So there is a sufficient vacuum
Sir,
moisture.
Agree, sir. If
sir, all the microbial species they can survive in very less water also,
Okay.
even in dry conditions. But they may not reproduce in that condition.
Oh, I see. Okay, good. So, but if it is a matter of a high vacuum, so there high vacuum means there is a, I think we suppose there is a no moisture. So it can't survive then. At a
So
certain
it
level
can't,
of vacuum, it can survive.
it might survive, but it can't reproduce, obviously.
Okay,
It might
okay,
survive.
good.
actually,
Good.
sir, because their growth, as Dr. Kamble said, he's right. They will survive, but still the vacuum will be very high. They will not survive, but they will not.
Yeah,
Yes.
right.
Yes.
Okay, good. Okay, so thank you so much. We improve our understanding about the biological science as well. Thank you so much, Dr. Dhankar, Dr. Kamble, especially Dr. Dhankar for being a resource person of this very enlightened, informative session. Once again, thank you so much for your time, your effort for connecting all of us. And at the same time, I would like to extend my thanks to the coordinator, Dr. Renu Chaudhary, and her team, Dr. Kamble, Dr. Sethi, and all other members who have supported the session, along with the faculty members of SoBAS and students.
Thank you, sir.
Besides, I would like to extend thank you to all the attendees, external faculty members, parents, whomsoever have attended this session. Thank you so much for your time and your support throughout. You know, we have organized this kind of, we prepared this kind of a series for the benefit of the society in large, not only for GD Goenkans, but also for the other students, faculty members, so that we work together and improve our scholastic understanding. Well, moving on, as you can see on the screen, there is a next session of SDP session. The speaker is myself. So, and the moderator is one of my very bright colleague and friend, Dr. Shasikant Gupta ji. I think Dr. Gupta must be here. Dr. Gupta will moderate the session, and I will speak about a scientific advancement towards peace and harmony post Second World War. So usually the science was developed from ancient time till the 20th century, and then it was actually the nuclear war devastated Europe, America, Japan, everywhere. So basically we believe that scientific achievement must be utilized for the peace, for establishing peace and harmony for the greatest benefit of mankind, for the benefit of nature. So that's what I will speak. I may request all the members, all the attendees to join the session and participate in the discussion. Thank you so much indeed. Once again, I wish you a nice and wonderful afternoon. Stay safe, stay healthy, and certainly stay at home. Still, we have to face the corona outbreak epidemic situation. Thank you so much. Have a very nice evening. And we will see you in the next session. Thank you.
Thank you,
Bye.
sir.
Thank you, sir.
Thank you to all coordinators who helped to conducting session smoothly. Thank you so much. Thank you to all participants.
Great. Thank you. Thank you so much. Take very good care. Bye.
Thank you, sir.
Thank you, ma'am. Thank you, everyone.
Thank you, everyone.